Yeah. Okay. Hello, everyone. Sorry. Hello, everyone. I'm Juan Lucas from Sydney Botanic Garden. I'm a horticulturist. I'm a partner investigator of on the NICE Hub, working alongside Professor Ho-Kyon Chong and the Dr. Ibrahim Salibi, both from the UTS. Right. And sorry for my pronunciation. And today we will present part of our project about applying and studying the effect of human urine fertilizer on plant growth. This is a 4-year project where each year we work with different plant species, which are grown in pots and fed with urine fertilizer. We compare the growth of each species using the urine fertilizer versus the commercial fertilizer. In our first year, we worked with the spearmint, and in this year, our focus was on annual plants. And because of that, we chose the hybrid Viola batrociana, which is a very common garden plant. You can see the picture of the plant with the flowers. And for this year of the project, we proposed the following objectives. Our first objective was to study urban fertilizer effects on annual plants growth compared to a commercial fertilizer. But also we decided to compare under which potting mix the plants will grow better. And our third objective was to investigate any growth inhibition parameters on the plants. And to achieve these objectives, we designed the following experiment, which took place in the nursery installation of the Sydney Botanic Gardens. Here's a picture with all the glasshouse. And this experiment consists in 2x2 factorial designs with 5 replications each. And we basically work with 20 plants that we potted up in 145 millimeter pots. And we used 2 different media to grow the plants. One media was equal part of peat moss and perlite, and the other media was 100% cocoa chip. You can see in that picture over there. And after we potted up the plants, we randomized the plants following a completely randomized design. And we fertilize the plants with the 2 different fertilizers once a week, with commercial fertilizer and the urine fertilizer, with a concentration of 200 ppm of nitrogen each. And we apply 100 mils of the fertilizer per plant. We collected our data on weekly basis of the diameter of the plants, the height of the plants, and the total number of flowers per plant. And we ran this experiment for a period of 7 weeks. And after we completed the experiment, we measured the fresh biomass and the dry biomass of the plants. And once we analyzed the data, we obtained the following graphs. In this first graph, we compare the plant's diameter for each type of fertilizer. On the left, you see the plants grow with the commercial fertilizer. On the right, the plants grow with the urine fertilizer, and it's no significant difference in the diameter between the 2 fertilizers. In this second graph, we looked at the plant's diameter across the 2 different potting media. The plants grow with the media consisting in equal part of peat moss and perlite, as shows in the right box, tend to have a larger diameter than the plants grow with the cocoa chip media. The next graph, we can see the number of flowers for each treatment. The first box on the left shows plants with commercial fertilizer and a cocoa chip media. The second box displays plants with commercial fertilizer and a mix of peat moss and perlite. The third box shows plants with urine fertilizer and a cocoa chip media, while the last box have plants with urine fertilizer and the mix between the peat moss and perlite. And as you can see, there is no clear difference in the number of flowers between the treatments. But in this last graph, where we see the fresh biomass weight of each treatment in grams, the second bar, which represents plants treated with commercial fertilizer and a mix between peat moss and perlite, stand out. Is that one with the orange circle. It shows an increase in biomass compared to the other treatments, meaning these plants grow more than the other ones. And in conclusion, from the data we collected, we can say that the highest biomass results were obtained using commercial fertilizer combined with the peat moss and perlite mix, which produced plants that were 30% bigger than the other plants under the other treatments. In terms of the plant diameter, those grow with the peat moss and perlite mix shows a slight tendency to be large compared to those grow with the cocoa chip media. However, when comparing the number of flowers per plant, as well as plant's diameter and height, we did not find a significant differences between the treatments. In addition, no growth inhibition was observed in any of the treatments. However, this is just an initial phase of our research. Our goal is to continue investigating and exploring the effect of human urine fertilizer on different plant species in the coming years. And this will help us to reach better conclusions. Finally, we would like to thank the Australian Research Council for their help throughout this project. We would also like to thank Yuan Song for providing the urine fertilizer. And finally, we want to acknowledge the in-kind support, effort, and time employed by the Royal Botanic Gardens CINO. Thank you very much.
Every speaker, please come to the stage. Very interesting, and the usual value chain, the discussion we have. So any question do you want to ask?
The question, I guess, on the gratis trial at University of Western Sydney.
Yes.
Yeah, the rate was 200 kilos of actual elemental N per hectare, yeah?
Yeah.
So that's a fairly high dose of nitrogen, which you probably wouldn't normally apply to a pasture, really. But yeah, I'd be interested to see what, if any, toxicity associated with pH change, because what was the actual dose of Jubal in the pots? Because at 200 kilos of nitrogen, that's like 66,000 litres per hectare of Jubal, which is a lot.
Yeah, so actually those things we have taken reference from UK grassland and the Australian grassland. They have recommended and they have already used 250 highest. So in the first, when we get the urine, we don't have any idea how much will be like highest and minimum dose. So just not taking 250, we choose 200 and 100 just normally to see, but we haven't just used that way the concentrated one. We just calculated by testing the urine 38 gram per liter, and then we make equivalent to that and dilute it, and then we give it to fertilizer and making exact amount with the mineral fertilizer as well. Yeah,
So it's the nutrient use efficiency too, like that. The nitrogen is 100% available as opposed to a mineral fertilizer where it's released in a much slower to the plant. So there's probably a nitrate toxicity factor going on there where you're giving the plant a big dose of 100% available nitrogen.
so yeah, we suspect maybe nitrogen toxicity, maybe some ammonium as pH is decreasing, so metal oxide toxicity, but I can't say because I don't have soil data, only I have plant, but soon I will, and that's why I simplify the dose now.
Yeah.
That exact dose will be good, which will be healthier for this plant species. So soon I will provide the dose with this experiment. So goal is to optimize the dose from this experiment.
It's really difficult, and I guess it's a question for you, if you fertilizer trials, it's comparing liquid fertilizers to granular fertilizers and their release patterns it's
Yeah.
very difficult to compare apples with apples. And
Yes.
I guess looking at the way that the release of that nitrogen occurs, you need to do a baseline in terms of how much nitrogen is being released over time of those granulars before you can compare that to a liquid that's going to give you immediate nitrogen response. So yeah, so it's a hard thing to do, but that's required to really understand what's going on.
You try to like, it's a one-time application.
Yeah, that's what we did, but this time I'm doing split and one time both. So the first experiment when I came, I just wanted to see, at least I wanted to find the boundary. So I found the boundary. So I'm minimizing and splitting it now and using also foliar application, two different application method.
Right, question. Oh, probably all three of you.
Yeah, it's good.
Do you actually measure, I know you have to measure the biomass and the new growth and the plant health, but have you looked at the root health?
I do.
Do you measure that as well?
I did it. Also with the leguminous one, I have counted the nodules.
Okay.
The good, not the good, but with the urable treated, the nodules was suppressed. There's no nodules with the urable one. But other mineral fertilizer, we have seen some nodules. But yeah, I have measured the root data, but as I said, it was something we wanted to check the boundaries. 200 kg was a bit higher for the pasture crops, but yeah, so overall we will soon give the good dose for just the pasture with the urable, but with 100 is a very good. With the urable 100 kg is really very good.
In my case, we have not done for that experiment, but we look at physical, how to say, checking on the roots. But in the next experiments, we will definitely do some root analysis as well as also checking of the overall biomass of that. But based on our earlier study, the roots is a lot more dense in the urine. Yeah,
Okay.
much. It's very difficult actually to separate with the vermiculite.
Yeah.
We have very difficulty to do that. It's just too much going around.
It is hard.
Yeah.
Yeah, when we measure the fresh biomass, we measure everything, the leaf and the roots.
Okay, third round, yeah.
So,
Again, the grass, but